Hi everyone, welcome back to your channel Agriclarity and Clarity for Sure. Today we'll be discussing important MCQs from the subject genetics and planting. Okay, so before proceeding into today's session, I would like to request every student watching this session so far to please go to our channel Agriclarity, where we have posted a separate playlist for PYQ analysis of all the subjects and we have given the analysis from the year 2021 to 2024. Whereas all the subjects of plant science, whether it be genetics or plant pathology, entomology, physiology, all these subjects have the analysis from the year 2017, right? So आपको इस analysis में you will get year-wise distribution, topic-wise distribution, then what are the frequency analysis of what are the frequent occurrence of these topics, and what are the topics to skip, and what are the topics you have to skim, and most probable and then predictability of high, moderate, and low probability topics. Okay, so this sessions or these videos are very helpful would be very helpful for you to have a targeted टू हैव योर टारगेटेड प्रिपरेशन राइट. सो मैं रिक्वेस्ट करती हूँ कि जो भी स्टूडेंट अभी तक हमारे इतने 2-3 वीक से हमने सारे प्रैक्टिस सेशन स्टार्ट किए हुए हैं, तो आई होप सारे जो भी सब्जेक्ट्स आपने अभी तक पढ़े होंगे या जिनके क्वेश्चंस आपने सॉल्व किए होंगे उनका एक बार आप पीवाईक्यू एनालिसिस जरूर देखें. इट इज बहुत ज्यादा लंबे वीडियोस नहीं है, लेकिन इट इज वेरी फ्रूटफुल फॉर ऑल ऑफ यू. ओके, सो बिकॉज किसी भी एग्जामिनेशन के आगे जाने से पहले इट इज वेरी इंपॉर्टेंट यू गेट द आइडिया ऑफ हाउ द क्वेश्चंस वे फ्रेम, व्हाट आर द टॉपिक्स, व्हाट आर द टॉपिक्स यू हैव टू नेग्लेक्ट. ओके, क्योंकि सारी चीजें पढ़ने होते नहीं, कुछ चीजें आपको नेग्लेक्ट भी करने हैं. देन ओनली यू विल गेट टू कवर ऑल द सब्जेक्ट्स, राइट? सो इसीलिए मैं काफी ज्यादा इस पे एम्फेसिस दे रही हूँ कि प्लीज गो एंड चेक आवर पीवाईक्यू एनालिसिस प्लेलिस्ट अवेलेबल ऑन आवर चैनल, राइट? Let's move on to the first question of today's session. First question of today's session is which of the following, yes, which of the following pair is incomplete? Wait a sec. Yes. Yes, which of the following pairs is incorrectly matched regarding the historical developments in the genetics? Okay, so again, here we are targeting the questions from the historical perspective. Okay, so which is very important questions वहाँ पे आया. अगर आपने PYQ analysis देखा होगा तो you know why I have put first question from the historic perspective, right? So option is W. Welder coined the term chromosomes, Bateson coined the term genetics, Jonasson coined the term gene, D. Varies coined the term meiosis, and C. Golgi who described the Golgi apparatus. So you have to choose the incorrect here. So answer is option D. Okay. Why? Because the term meiosis was coined by J.B. Farmer and J.P. Murray in 1905, not by H.D. Varies. Okay. Please drop your answers. What discovery was done by H.D. Varies here? Okay. If you know this answer. Okay. Moving on to the next question, which of the following statements about the Mendel's law is not correct? Again, we have to choose not correct because not correct question is like they assess your concept clarity. Okay, they assess how clear you are with the basics of your particular subjects. Okay, so that is why I have given not correct because correct वाले questions के आपको बहुत सारे जगह मिलेंगे, लेकिन not correct is the one because you have to face some kind of difficult questions. Okay, be ready for the difficult questions from in your examination, right? So the first option is law of segregation is also known as the first law of Mendel. The law of independent assortment leads with the inheritance of single trait. Law of dominance explains the appearance of only dominant traits in F1, and the law of segregation was later supported by discovery of meiosis. And lastly, law of independent assortment is based on the dihybrid cross results. The answer here is option B, which is false, because independent assortment deals with the inheritance of more than one trait, not a single trait. Okay, if you are familiar with these three important Mendel laws, law of segregation, important independent assortment, and law of dominance, you would have easily eliminated the options, right? Next is question 3. Which of the following scientists discovered the crossing over and recombination in Drosophila? Again, one of the important topic from the genetics. Okay, C.E. Correns, W. Johnson, T.H. Morgan, H.G. Muller, and W. Bateson. One of the easiest question. So answer here is C. Okay, T.H. Morgan discovered the crossing over and recombination in the Drosophila, right? Moving on to the next question. You have to again choose the not correct about allele related terminology, one of the important unit of genetics, or we can say gene, right? So here isoalleles are completely different alleles found on non-homologous chromosomes, right? Then pseudo alleles are closely linked to genes separable only by rare crossing over. Is it correct or not correct, right? Then multiple alleles refer to more than two alternate forms of gene at the same locus, and Nast's allele was first called factor by the Mendel. Okay, so the answer here is option A, means only first was the incorrect here. First, why? Because isoallele are slightly different alleles at the same locus, not a homologous. Okay, not a homologous. That means they are heterologous chromosomes, right? That is why it was incorrect here. The rest are correct because pseudo alleles are tightly linked with the genes separable only by rare crossing over. Then multiple alleles exist at same locus, and Mendel called the alleles factor, right? Moving on to next question. Which of the following combination is correctly matched with the respective definition? You have to choose the definition and their characteristic, right? First is codominance. One allele completely masks the other. Second is lethal gene causes death when in heterozygous condition. Then epistatus. One gene masks the effect of another. Pleiotropism, a gene affects only one trait, then cytoplasmic inheritance, character passed through male parent. You have to choose the correct match here. Okay. So answer is C. Epistasis, one gene masks the effect of another. Okay. So here is it is correct. Okay. Rest, I hope why other options were incorrect is very clear to all, because these are very simple definitions. You have to learn these simple definitions because the questions are straightforward here, right? Next is, which of the following statements regarding polyploidy and aneuploidy is not correct? One of the important phenomenon in plant breeding, right? Polyploidy and aneuploidy. So here we are asking triploidy. Okay, triploidy represents the three complete sets of chromosome, which is 3n. Okay, monosomics have one chromosome less than diploid sets, so it is denoted by 2n minus 1. Then tetrasomics is denoted by one extra chromosome, 2n plus 1. Double trisomics contain extra chromosomes like 2n plus 1 plus 1. Okay. And the pentaploidy refers to 5n. So you have to choose the not correct here. Okay. So the not correct answer would be C. Okay. Tetrasomic actually has an extra pair, homologous chromosome, not 1. Okay. So that means the answer would be 2n plus 2. Okay. So that is why it is 2n plus 2 is equal to 4, right? So that is why it is tetrasomic. Okay. It is very easy. You can just, from the name itself, you can eliminate the options, right? Triploidy is 3 and monosomic is 1. Tetrasomic, there is no findings of the 4 chromosomes, right? Then double trisomics. So trisomics means 3, then double is 1, 1. Okay, so that is why 1, 1. Then penta is 5. So it was a very easy question, right? Moving on to the next question, which of the following correctly matches the type of mutation and its description? Mutation, one of the recurring topic in genetics and plant. If you have come from the, I mean, if you have had watched our session, PYQ analysis session, then you must know that why I have given the mutation question here, right? Then you have to choose a definition and with the description, right? Then option is deletion, repetition of segment of chromosome. Duplication means exchange between non-homologous chromosome. Then inversion means rotation of chromosome segment through 180-degree. Then translocation means loss of chromosome segment, and substitution means addition of nucleotides in DNA sequence. So you have to choose the correct match here, not incorrect, correct match. So the correct answer is C. Okay. C is the inversion, changes the degree or gene order by 180-degree rotation. Okay. Rest were wrong. Okay. Repetition is not related to deletion. Then duplication is like they are in one extra number, right? Then transcoloration is they have to do something with the placing, okay? And substitution is when it is substituted with other thing, right? So in this way, you have to just by the name itself, by the generic name itself, you can guess the type of mutation, right? Next is, which of the following gene subdivisions is incorrectly matched with its function? You have to choose again here incorrect matching of gene subdivisions with its function. Okay. Then if you have, I mean, let's see. Let's see, then I'll explain. Okay. Then recon, which is meant by the smallest recombination unit within a gene, muton, smallest unit capable of mutation, cistron, smallest functional unit within a gene, a fundamental biological and hereditary unit, then muton, largest element within a gene controlling phenotype. So here the answer for the incorrect is E. Okay. Why? Because muton is the smallest unit, not the largest. Okay. So that is why it is wrong here, right? Next is in the lag operon model, which of the following correctly represents the sequence and role of gene. Okay, so you have to choose in what sequence the lag operon model conducts its phenomena, right? So operator structure, as you can see in the options, the correct option here is option C. Okay, regulator, promoter, operator, then it go on for the structure, right? So why? Because regulator leads to repressor synthesis, then promoter leads to mRNA initiation, then operator controls the process, then structural leads to protein synthesis. So in this way, the lag operon model is conducted. Okay. So you can see that there are very few topics which are important for genetics. Okay, you need not to do all the topics available in your subject. So you can see how the questions vary from, and they are touching every topic of plant breeding, right? Let's move on to the 10th question of today's session. Which of the following combinations of gene classification based on effect of on survival is correct? Okay. Again, we have to move on to the gene, types of gene. Okay. Then the option says that lethal gene causes death in the heterozygous state. Okay. Semi-lethal gene causes death of more than 50% carriers. Then subvital gene causes death of all carriers. Vital gene causes complete loss of viability, and all of the above are correct. So the answer here is option B. Okay, option B is correct because it kills semi-lethal kills more than 50% of carrier. Rest are wrong, right? Because lethal, I mean, it is for if you are familiar with the lethal gene definition, it is caused. I mean, it causes death in homozygous state also. Okay. Next is which of the following genes are correctly matched with this classification based on the sex linkage? Okay, sex inheritance and sex linkage, again, one of the important topic of genetics and plant breeding. Okay, so here the sex linked genes is found on autosomes. Sex limited genes are expressed in both sexes equally. Sex influenced genes expression depends on sex of individual. Then sex linked genes found in cytoplasm or all of the above. So you have to choose the correctly matched sequence here. Okay, correctly match option here. So answer is option C, which is sex influence genes vary expression by sex. Like the example is baldness. Okay, baldness, or you can say the ear hairs present in the male. Okay, so that is fine. Next is which of the following statements about mutation effects is not correct? You have to choose the not correct option from the topic mutation. Okay, so lethal mutations improve biological fitness, subvital mutation reduces the survival chances, and subvital, supervital mutations may improve the adaptation under certain conditions. So answer here is option A. Only A or we can say only one is not correct. Why? Because option one says that lethal mutations cause, I mean, lethal mutation leads to death, not to any kind of biological fitness. Okay. And subvital reduces the survival, and supervital improves the adaptation process in the entity, right? Next is which of the following correctly differentiates the primary and secondary introduction in the plant breeding? Okay, so you have to choose what are the characteristics which differentiate these two phenomena. So first is primary introduction involves modifying introduced genotypes through hybridization before the cultivation. Secondary introduction involves further selection or hybridization of the introduced genotype before the use. Both types are identical in their adaptation process, or secondary introduction does not involve any genetic alteration. So answer is B option, which is correct here. Why? Because secondary introduction modifies or uses the introduced material in hybridization to obtain a superior types before its actual use. Okay, so that is why the secondary, in this way, the secondary introduction differs from the primary introduction in plant breeding. Okay. Now, penultimate question of today's session. In which of the following cases is the back cross method most suitably applied. Okay, so a high yielding variety from the land races, improving a high yielding variety by adding one or two specific traits, developing synthetic varieties for general combining ability, and producing F1 hybrids with maximum heterosis, and lastly, developing polyploids for seedlessness. Okay, so you have to choose where you can successfully apply the backcross method here. Okay, so option is B, because backcrossing transfers one or two more desires, like you go for disease resistance. Disease resistance quality in your particular variety, you go for the backcross method, okay, to incorporate into a superior cultivar, right? And rest were not were the most suitable application of backcross method. Okay. Now the last question of today's session: which of the following is not correctly matched? You have to choose. Again, heterosis topic. Then you have to choose the what is which is not correctly matched. First is heterobeltosis, which is superiority of over beta parent. Average heterosis, superiority over mid parent. Negative heterosis, superiority over commercial variety. Or economic heterosis, superiority over standard commercial variety. Okay, so the correct not correct match is option C, which is negative, which means why? Because negative heterosis means inferiority, not superiority. And we are saying there it is superior. Okay, so that is why it is wrong. Okay, from here onwards, I have given 5 questions for your further practice. Okay, try to answer these questions in the comments below. Try to pause for 2 or 3 seconds and then read the question and try to answer these questions in the comments below. I will be, checking the answers given by all of you in the comment. And if there is anyone having any problem with the today's session or any topic you are not familiar with in today's any topic you feel that was not done by you from your perspective very good, so please let me know those topic as well. Okay, I'll try to come up with the video to have a better to have to to have a better understanding from your end. Okay, to get better marks in this subject, right? So this was it, and don't forget to join our Telegram channel where we are always posting questions from various topics, let it be horticulture, agronomy, or soil science extension. Every day we are posting 10 MCQs for all of you, matching the exact examination pattern. Okay, so you should join the Telegram channel. As you can see, we are getting very good number of views there, and many students are answering it very well. Okay, so try to join the Telegram channel as well. And that was it. If you feel any kind of doubt, any confusion, any topic you feel that you want us to cover, please let me know in the comments. And don't forget to answer the questions which I have given for you for the practice here. Okay, so try to be active, try to involve in these kind of videos. Okay, it is for your benefit only, right? And if you feel so that these questions were any at par of the IBPS AFO examination, please let us know. Please let us know any suggestion from your end elsewhere. And please check our test series, which are available at a very minimal cost at C4S courses. The website link is given in the description below. Okay, so that was it. Thank you and have a nice day.